Good afternoon. The topic of my poster is characterizing hepatocellular carcinoma inoculated in mice by fluorescence lifetime measurements. Liver cancer remains one of the most commonly diagnosed cancer and the fourth leading cause of cancer death. The problems of this type of cancer are the difficulties of early determination and rapid growth. That's why it is relevant to make the diagnosis faster. Currently, cancer diagnosis requires histological and cytological analysis of tumors. Samples are usually obtained by puncture biopsy. Despite many advantages, this procedure does not provide real-time information on tissue state. Another problem is the probability of taking non-diagnostic biopsy samples, which often leads to a second biopsy and increases the duration of treatment. The development of optical biopsy methods for standard procedures of minimally invasive surgery may offer a solution of the problem. Optical technologies seems to be promising for improving biopsy needle targeting and real-time in vivo monitoring of molecular and morphological structure of biological tissues. A number of optical methods are known to be applied for small diameter needles. In particular, fluorescence methods are widely used to assess the metabolic state of biological tissues. Several endogenous fluorophores have decent fluorescence spectra and are actively involved in cell metabolism. One of them is nicotinamide adenine dinucleotide coenzyme of mitochondria, or NADH. NADH fluorescence intensity and lifetime decay function bear information on its concentration and biochemical interaction with surrounding substances, which is directly associated with the metabolic state of the cells. The changes of these parameters can indicate cancer formation. Therefore, the aim of this study was to integrate fluorescence intensity and lifetime measurements into the optical needle probe and to test its capabilities in an animal model of liver cancer. To compare the fluorescence parameters of healthy and malignant tissue, the tumors were modeled in laboratory mice. The cells of hepatocellular carcinoma were inoculated into the middle lobe of the liver of BDF mice. The measurements were carried out 4-6 weeks after the injection. To measure both fluorescence intensity and lifetime, we used a time-correlated single photon counting system by Becker and Hickel. It included two photomultiplier detectors, 375 nanometers laser, and a monochromator to excite fluorescence, and 365 nanometers. The laser source and the detectors were connected to the specially designed fiber needle probe. Its distal end has 1 millimeter diameter, and it is designed to be used through 17.5 gauge standard needle with a 20 degrees bevel. The optical measurements were followed by standard histological study to confirm the stage of tissues for processing the data. This slide shows the results of measuring average fluorescence intensity. Fluorescence intensity increases in both intact liver and malignant tissues, with a slight prevalence in tumor tissues compared to the control group of mice. The values of both lifetime decay components significantly decrease in malignant tissues compared to intact liver. It can be observed that these components are low in control group as well. While the amplitude contribution of fast component is high in all the types of tissues, the tumors demonstrate significant increase of fast component contribution, so its value is almost 3 times higher than the one of slow component. Taking into account the previous slide, the obtained data shows the increase of metabolic activity in mice with inoculated tumor. However, the ratio of fast and slow components contribution indicates that glycolysis mostly dominates in malignant tissues. Intact tissues has increased rate of oxidative phosphorylation, which may be affected by the presence of tumor in the liver. The conclusions for the study are following. The obtained data showed that violations in cell metabolism have led to changes in bound and unbound NADH ratio, which were observed with the time-correlated single photon counting. However, the wide range of values recorded can indicate not only the fact of malignant cells growth, but also the stage of tumor progression and individual characteristics. Therefore, it is interesting to carry out further studies of these features. In general, this study shows that further study of fluorescence lifetime parameters seems promising for development of additional diagnostic criteria to introduce them into algorithms of minimally invasive diagnostic procedures. The study was supported by the Russian Science Foundation. We also would like to thank Dr. Olga Morozova from Blakhin Russian Cancer Research Center for the animal models. Thank you very much for your attention.